This is Quentin and Ashley's iMovie, and we will be showing you how mitosis and meiosis work using different types of candy. In prophase 1, the chromosomes begin to condense and the homologs begin to pair along their lengths. Crossing over also begins to occur. The chiasmata is where the genetic information is swapped between the chromosomes. Also in prophase 1, the spindle is beginning to form and the nuclear envelope is beginning to break down. In metaphase 1, the pairs of homologous chromosomes begin to line up at the metaphase plate. Both chromatids of the homolog are still attached to the kinetochore microtubules coming from each pole. In anaphase 1, the pairs of homologous chromosomes begin to separate, and the homologs move toward opposite poles. Sister chromatids remain attached. In telophase 1, each half of the cell now has a completed haploid set of duplicated chromosomes, and in each chromosome there are two sister chromatids, which include regions of non-sister chromatid DNA. Cytokinesis happens at the same time of telophase 1, in which the forming of two haploid daughter cells occurs. In meiosis 2, the sister chromatids will separate during another round of cell division, which will result in 4 haploid daughter cells. In prophase 2, the spindle apparatus forms. Next, in metaphase 2, the chromosomes are lined up at the metaphase plate again, just as seen in mitosis 1. The two sister chromatids are no longer genetically identical due to the crossing over that occurred in meiosis 1. And the kinetochores of the sister chromatids are still attached to the microtubules and are beginning to extend from opposite poles. In anaphase 2, the proteins that were holding together the sister chromatids together at the centromere begins to break down, which allows the chromatids to separate. The chromatids then move toward opposite poles to individual chromosomes as a result. In cytokinesis, the division of one parent cell results in 4 unique daughter cells, each containing a haploid set of unduplicated chromosomes. During cytokinesis, 4 unique haploid sets of daughter chromosomes. In interphase, the nuclear envelope encloses the nucleus as a whole. 2 centrosomes have been created by the duplication of one centrosome, and the chromosomes have just been duplicated during the S phase of interphase. Next, in prophase, the chromatin fibers become very tightly coiled, and the nucleoli disappear. Each duplicated chromosome consists of 2 identical sister chromatids joined by their centromeres. The mitotic spindle begins to form, which is composed of both the centrosomes and the microtubules that come out from them. In metaphase, the centrosomes are now at opposite poles of the cell. The chromosomes then line up at the metaphase plate, and the kinetochores of each sister chromatids are attached to the kinetochore microtubules coming from each opposite pole. The cell then elongates as the non-kinetochore microtubules lengthen. The cohesin proteins are disconnected, which allows the 2 sister chromatids of each pair to move apart. The two independent daughter chromosomes now begin to move toward opposite poles. In anaphase, which is actually the shortest stage of mitosis, lastly, in telophase, the two daughter nuclei form in the cell, and the nuclear envelope reappears, while the chromosomes become less condensed. The division of one nucleus into two completely genetically identical nuclei has now been completed. At the same time, cytokinesis is taking place, in which the division of the cytoplasm occurs, which results in two daughter cells.